up first, we have Morgan Irons, who's going to be doing the Deep Space Ecology 3-zone model.
Thank you. Hello, everyone. Like what was said, my name is Morgan Irons. I am the Chief Science Officer of the company Deep Space Ecology. I helped found this company last year in May, so we've just passed our 1-year anniversary. Good time to celebrate it, yeah. Yep, and so I will be speaking on closed ecological systems and what my company is doing with that, as well as some of the research that I performed while I was attending Duke University. So what exactly are closed ecological systems? The experiment that may came to mind immediately is Biosphere 2. That was a closed ecological system. Didn't work out, but we learned quite a bit from it as well. And so when you think about closed ecological systems, you can think of them as a habitation module on Mars, on the moon, or something as simple as just a little bottle that you see elementary school students, middle school students create, where you get the soil, the water, and the plants, you put them in a liter bottle and seal it up, and now you have your own little terrarium. But when it comes to larger scale things like what we would see with habitation systems, closing off an ecosystem leads to a bunch of problems. And so some of those problems that come with that include atmospheric fluctuations and increasing volatile organic compounds with the introduction and growth of plants and other organisms. And any engineers in the room might say, well, why don't you do an intake system, use the atmosphere from outside of your closed system to help that fluctuation and to help stabilize it, or use a scrubber system? So that's a potential solution to this problem. Getting a little bit more complicated, you can also have out-of-control biological activity of microbial populations as they start to rapidly decompose organic material introduced to the soil, which can then affect the concentration of carbon dioxide in the atmosphere. So something that happens in the soil can also affect what happens in the atmosphere, showing that there are different ways in an ecosystem that things can be manifested, which is important to think about because ecosystems don't work in an engineered step-by-step system. They're very fluctuating, and they do a lot of rapid changes with just little introductions to things. And then continuing on, if my thing works, let's see. You have the lack of essential elements present in active circulation due to the new environment, such as on Mars or on the moon, not having nutrient available in the chemical form that the plants can take up. What comes to my mind when I think about this is the scene from The Martian and NASA releasing a list of the nutrients that you would find on Mars that are available there for plants to grow. However, what they don't tell you is whether they are bioavailable for those plants to take up. So it's important to consider not only what elements that we'll find on Mars, but also how we would be able to change those nutrients into a form that plants can take up. And then you have degradation of soil fertility with reduced circulation of energy, nutrients, and materials. With a closed ecological system, you have it closed off from the outside environment, which is different from the ecosystems that we have here on Earth, where you can have animals and water flow through the system, bringing with it different nutrients, different things that would allow the ecosystem to stabilize and be resilient. And then, of course, the list goes on and on. And so the important takeaway from this list and from past experiments is that the problems that we see, it is hard to predict what disruptions will occur and the pathway that they will take when the reaction takes place. And so with my company, what we do is look at these problems, and we developed our own model for a closed ecological system called the 3-zone model, which I introduced at last year's conference, which is now a patent-pending invention. And one of the things that it takes into account is how ecosystems form here on Earth, a process known as ecological succession. How do you get from bare rock to a climax community that you see outside today? By following this process and understanding this process, you can avoid the previous challenges that I listed on the previous slide by allowing the ecosystem to naturally form the feedbacks and the cycles that it needs to form the ecosystem. But how can you go about doing that? And so that's where my thesis work at Duke University came in, where I was, the title of my experiment was The Development of Ecological and Environmental Mechanisms in Sterilized Mars Regolith Simulant to Drive Primary Succession in the Establishment of a Closed Ecological System on Mars. And so with this experiment, I was pretty much focusing on understanding and applying ecological theories and principles to develop and manage a quasi-closed agroecological system for extreme and changing environments here on Earth, as well as in space life support systems. And so the foundational research I performed looked at understanding how biogeochemical cycles and feedbacks may be developed in a sterilized, degraded soil or a regolith condition, pretty much landscapes that are prior to a primary ecological succession occurring, that would later be used for agriculture and be expected to provide ecosystem services to the organisms living in the immediate area. So this initial research led me to investigate through experimentation the trade-offs between using biodiverse species of plants and microorganisms and using different combinations of pretreatments to the regolith, such as the presence or absence of grade A biosolids for a fertilizer use, and then different combinations of organic litter, tilling, and watering techniques. The results looking at whether there are statistically significant associations between different soil amendments and the plants and microorganism biomass production. So to get into a little bit of the background, this research was done at the Duke University research greenhouses. A constant temperature was maintained. The daylight hours were maintained as well through natural as well as artificial lighting. And this experiment lasted from June 1st, 2016 to December 20th, 2016. And the different pretreatments that I was looking at to adding to the regolith was an initial water content condition, organic litter presence, microbial inoculation with biosolid application, and then different plant combinations. And here you have pictures of those different pretreatments. So you have the Mars regolith simulant, which I got from SciArt Industries out here in Napa, California. And it's pretty much a combination of basalt, sand, and dust. And then the different plants that I was using included golden peas, Sonora wheat, broccoli, rapeseed, and French sorrel. The rapeseed and French sorrel were used as an organic litter content where they were grown and then would be tilled down into the soil to start creating that O horizon or that organic horizon that's found in soil profiles. And then I used microorganisms, including mycorrhizal fungi and rhizobium bacteria. Mycorrhizal fungi help with root establishment and uptake of water, while rhizobium bacteria is that nitrogen fixture that helps take that nutrient and turn it into a form that plants can uptake. And then, of course, I used biosolids from a local water treatment facility to use as a fertilizer, and it was not pleasant at the beginning to use those. And so the hypotheses that I was looking at for this experiment included the addition of a rhizobium bacteria, mycorrhizal fungi, and biosolid pretreatment increased the growth and establishment of plants growing in a Martian regolith simulant. And my second hypothesis, the addition of the pretreatments acting as the tools for primary succession to initiate over the experimental time changes the chemistry of the soil to reflect the development away from a protosoil. Protosoil being that initial soil content that you start to have as microorganisms and those initial pioneer species enter the ecosystem that's going under ecological succession. And so here's the experimental design. I had 10 replications that were happening with a control group, and within each replicate was 12 different combinations of those pretreatments. So it ended up being 120 pots being tested with additional 12 pots for the control group. And you can see a picture of the experimental space right over here. I did have help setting this up. I did not do it by myself. And so to go into some of the results, you can see right here with this chart, this data distribution shows the biomass production results for, that were produced across the pots. So the top chart shows the biomass produced when the microbial inoculation in biosolid pretreatment was present versus absent in the pots. The middle one shows the biomass produced in pots with peas and broccoli present versus pots without peas and broccoli. And then the bottom one shows the biomass produced when the Mars regolith simulant was initially wet versus initially dry, which I forgot to mention previously, the difference between the orange and the blue. The orange is with pots that did not have any water initially added. It was just dried, it had been sterilized, so that any microorganisms that were living in it were dead. But there were no water amendments to it at the beginning before the plants were planted. And then the blue is for the pots that had water initially added to it, went through a tilling treatment, and then had the plants added to it. And so with this, when I ran a statistical analysis on it, I saw that the significant pretreatment in pots growing wheat, so the pots that just had wheat growing in them, and then pots that had wheat, broccoli, and peas growing together, the microbial inoculation in biosolids application was the most significant association with biomass production, which at the beginning I was very confused because I would think that since peas are a legume, they would interact more with the microorganisms since they have a symbiotic relationship in that regard. So I went further and ran a soil respiration test where I created incubation chambers for the soil and measured the soil respiration rate, or the CO2 percentage, over a 56-hour time period. And the blue shows the pots that were given the microorganisms in biosolid pretreatment, while the orange did not have the pretreatment. And as you can see, the pots that had the microbes and biosolids had a higher rate of soil respiration over time than the pots that didn't. So it was showing that the microorganisms were being established and actually surviving, which is what you always want. And so this gave me a little bit more insight into potentially why wheat was having a better establishment rate with the microorganisms and thus producing more biomass. And the more I thought of it, the more it made sense, because wheat's going to have a larger, more fibrous root system than you would have for peas and broccoli. And so the microorganisms would be able to better establish onto these roots and create nodules and live a healthier life than with peas and broccoli that have the smaller root systems. And then moving on, I also looked at the results for carbon to nitrogen ratio tests, looking at how the biosolids were used with the combination of microorganisms. And so, keeping with the color scheme, blue has the microbiosolid treatment, orange does not, and the black represents the Mars regolith simulant control group that had no pretreatments whatsoever put into it, just to show the baseline that we're looking at. And so what you can see with the blue lines, which were the pots with the pretreatment, they had lower levels of carbon to nitrogen ratio test. The nitrogen being measured being the nitrate and ammonium elements, well, compounds, which is the form that plants take up when they're taking up nitrogen. And so we can see that the microorganisms are changing the nitrogen into a form that the plants can take up, which shows a healthy microbial population and a healthy beginning of an ecosystem. And then looking at pots that were growing peas and broccoli, these pots, it can be pretty confusing, so I'm These are the pots that have the mixture, peas, broccoli, and wheat growing in one pot, and then pots containing just peas and broccoli. And so with this, the significant pretreatment result was having the presence of wheat in the pot produced more biomass production, which immediately makes sense to me, because when you're looking at wheat versus peas and broccoli, wheat's going to produce more biomass naturally, and the peas are going to be smaller, so they're not going to produce as much. So that makes sense. And then a note to the side is the microbial incubation, inoculation and biosolids application was close to having a significant effect. The significant level being P equals 0.05. If it had been below that level, it would have be considered a significant association. But since it was a little bit above, I couldn't say it. But I decided, again, to look at the incubation and the carbon-to-nitrogen ratio test to see what's happening on the soil level. And so with the soil respiration test, you can see, let me do that. Okay, so the blue is the pots growing wheat versus the orange, which is the pots not growing the wheat. And you can see that in each case, everything's going in a positive trend, more so than the previous one, since all of the pots also contain peas, which is a legume, which would affect the soil respiration rate of the microorganisms. But again, wheat has that higher association with a higher respiration rate because, again, the root systems are larger, more fibrous, helps establish those peas. So when we're considering what plants to grow in a closed ecological system, you're not only considering, oh, I need this plant to make food, I need this plant to do this other thing that I will need as a human, but you also have to consider what those microorganisms will need, what plants will react with the soil in a certain way. So we're not only talking about a human-centered model, but a model that has everything on level with what's important for the ecosystem, what's important for humans, how can we create this system that's in balance. And then with the carbon to nitrogen test, it has similar trend to what we saw before with the pots containing the microbial biosolid pretreatment having lower CN ratio values, since the microorganisms are converting that nitrogen into a form that the plants can use, which is what this is measuring, while the pots that did not contain the microbes and biosolids were similar to a regolith that just didn't have any pretreatments added to it. And so, saw this picture previously, but this is how the regolith looked when I was first starting off. Pretty much as soon as you added water to it, it would turn into cement, which was not fun when you had to go in and till it, and I just had a spade. Ooh, I gained muscles from that, that's for sure. So you can see that when I went in with the spade, it would come off in huge chunks, and you would you would have to hit it. But then, at the end of my experiment, it looked completely different, which I jumped for joy. And so you can see here that it looks more like a soil that you would find here on Earth. So with the different pretreatments I was using, it showed how important it is to consider those things instead of simply just throwing the seeds into the soil. You have to consider how soil develops from just a sand and dust-based regolith. And then, depending on what pot and pretreatments were in there, let me step over here so you guys can see better. The regolith had different colors that it came off with the different chemical composition of the regolith, depending on if it had biosolids in it, microorganisms, the different plants that were being used. So that was also pretty fascinating. And so, to start with my conclusions, when it comes to ecological succession, my experiment showed very similar trends that have been seen in previous experiments dealing with primary succession, where the carbon to nitrogen ratios and respiration results show a steady and quick establishment. And then there is fluctuation in biomass with competition with those data distributions that I had when I was first introducing each of the results. They had wide arrow bars. And that shows that just when you're adding these organics to the environment, there are going to be plants that do very well, and there are going to be plants that do not so well. And so there's going to be initial fluctuations in that biomass, which is similar to what ecosystems go through when they're first forming. And it's similar, these are similar results to Chaplin, who did the Glacier Bay experiment, very famous foundational experiment, as well as other experiments like what's been done in Ganga Mountain in China. And there are a lot of papers that you can look up that do similar experiments as well. And then what made my experiment unique compared to other experiments looking at ecological succession is that the regolith I was using started off completely sterilized, which is not necessarily how ecosystems here on Earth formed. There may have been initial microorganisms in there, but when it comes to being on the moon or Mars, since we don't know whether there will be microorganisms in that regolith, it's safe to assume that we should start our experiments in sterilized conditions. And this creates a very different aspect to what we see. So with the absence of microorganisms in the regolith, I saw little to no competition with the microorganisms that led to that fast establishment of those microorganisms that I introduced into the regolith. And these are similar results to this study that was using phosphate solubilizing bacteria and azobacter in sterilized soil. And this is one of the only experiments on the internet that you can find on the internet or talking with professors that have been done in sterilized soil conditions. So this is a new field of study looking at sterilized soil conditions and creating an ecosystem from that. So more work needs to be done in that regard. And then the implications for such an experiment. Just being able to expedite biological feedbacks and cycle formations. In ecological succession, what usually happens is an ecosystem forms over decades to hundreds of years. Since we don't want to wait that long to live on Mars, we have to figure out a way to expedite this ecological formation. And so being able to do experiments like this where we're looking at how can we manually add things that the ecosystem needs and let it go and be able to get immediate results will help us further look at how can we establish these agricultural systems on Mars and on the moon or anywhere we go, really. And then better understanding the formation of ecosystems. In the biological and environmental science fields, we've made a lot of progress when it comes to understanding how ecosystems work, but we've pretty much only scratched the surface. There's still a lot of contention about what exactly happens on the foundational levels of how ecosystems work. And then increasing stability of a quasi-closed ecological system. With the problems that we saw previously with closing off an ecosystem, we need to consider how to stabilize it through allowing the ecosystem to form naturally, if not expedite it a little bit, so that we can stabilize it and fix some of those problems that have been seen in previous experiments. And then just to mention some uncertainties that came with my experiment, completely honest about everything. 30 of the pots that I was using were not comparable to the rest of the pots because of time situations where those pots were not ready to be analyzed. And then the root systems were not recovered for any of the plants besides looking at some of them to make sure there were microbial nodules, because when you went up to take that plant out of the regolith, it was still pretty cement and compacted-like. So as you can see here, pretty much the only root system that survived were the main tap roots and not the fibrous endings. Those stayed in the regolith, and it was nearly impossible to get everything out. And then this was an open, unsterilized location. I was doing it in an isolation zone of a greenhouse. And so I was isolated from other experiments, but the entire area wasn't sterilized. It wasn't completely closed off. So there was that as well. So what does this mean for habitation in space? There's still a lot that we have to learn. There's still a lot that we don't understand. My experiment gives me a little bit of hope when it comes to being able to do this in an extreme environment. But even with that hope, we have to think about the risks that still come with having a closed ecological system in any environment. And then how will we mitigate that? And so the problems of in situ resource limitations, a non-existent economy, an extreme space climate, and extremely long and expensive supply chain risks limiting human expansion and settlement anywhere outside of Earth increase the difficulty of developing such a habitation system at all. 2 minutes. With this deep space ecology, we developed a 3-zone model, which is a patent pending model, to help solve these problems. And so with the current space market trajectories, how are we getting to Mars? What's that road? And what's the greatest risk? In the space market, we have the ISS reaching its end of life. We have other nations entering the space industry. We have plans. We have people like Elon Musk saying that we should use the moon first to stoke public interest and provide opportunities such as for satellites and more Earth observation. So there's a lot going on in the space industry right now. And so you may say it would be easier to build such a system on Earth, such as in a location such as drought-stricken Somalia. And you may be right by this. But when you focus on the differences in gravity, atmospheric pressure, light levels, temperature, radiation levels, and the length of supply chain, beyond these differences, the food security problem and the Mars food security problem are actually very similar. With dwindling agricultural resources, extreme climate events affecting our regional food supplies, political conflict, large conglomerate farms affecting our small farmers. We need to find a way to create more sustainable solutions for growing food and feeding people on Earth, just as we need more sustainable solutions for growing food in space. And so with my company, we help solve food sustainability in space by concurrently looking at how to solve food security on Earth, looking at those similar problems and how we can spin that into space exploration. And so there are various Earth market needs, and I have to speed up a little bit because my time's running out. But we have needs of humanity, we have needs in the agricultural industry, and then we have food security and resilience needs as well. And with those needs, my company, Deep Space Ecology, we are finalizing arrangements to build our small-scale mock-up and prototype in which we can perform Earth-based experiments and technology tests for the Earth market as well as the space market. We're working with STARS and Alpha Space on experiments for the ISS. We currently have a team of 30 engineers and scientists working on system development for ISS experiments and for the moon. And then the dual purpose potential of the systems being developed will allow us to launch products into the Earth market as we move towards space deployment. People who have need to get reconnected to their biomes without moving from their country. There are also market appeals for buying from local food growers and providing food solutions for isolated communities that have limited capability. Lastly, there is a need for community resilience against climate change and natural disasters. These markets and the current trajectory of the space market give us plenty of opportunities for expanding the vision of humanity in a positive direction, and we at Deep Space Ecology want to be part of that vision. Thank you so much. Do we have time for questions, or do we?
You could probably do like one while we get the next person.
Okay.
How are you funded?
So we are currently in the process of looking for investors as well as letters of intent from investors as we move forward. Actually, the CEO of Deep Space Ecology is here. Yeah, we're in the process of pitching.
You have 30 engineers working.
Yeah, they are all working on spec.
Ah.
Yep, so they're all volunteers, and they're all amazing people.
Assuming with and without modest genetic manipulation, what's the minimum concentration of nitrates you might need to be able to do that its own fixing?
So, I had that number.
I mean, there's two different.
Yeah.
There,
Yeah. I would say.
I mean, is Mars near it?
Hmm?
Is Mars near that limit? Probably.
They definitely don't have enough to support an agricultural system that we would need to support a crew of at least 8 people. So you would definitely have to bring those amendments with you for a fertilizer.
There are enough in the atmosphere so you can find the atmosphere concentrated.
Yep.
But if we do modest genetic manipulation, might you think it might be able to allow plants to eventually grow on the surface once the air pressure is high enough?
Yep. I think that we should have.
Time now. Any further questions, please say here afterwards.